So far, we have discussed about the embryo development, and this embryo development is occurring in the embryo sac. And this embryo sac, in turn, it is present inside the ovule. And this ovule, after the fertilization, it is resulting in formation of seed. So the next post-fertilization event is formation of seed. So we are going to learn how this seed is formed and how this seed is going to be developed. What are the various types of seeds we are going to understand. So this seed, before understanding the seed structure, we are going to understand the structure of ovule because uh, the same ovule is after the fertilization forming the seed. So this ovule, if you see the structure of ovule, we have to remember some of the structures of this ovule. Those are integuments. Integuments are outer coverings, and these integuments are envelop. These integuments are they are enveloping the ovule except a area where it is forming a opening, and this opening is called as micropyle. Micropyle is the opening where the integuments are absent, and this micropyle is allowing the pollen tube to enter inside the embryo sac. And inner to these integuments, they have this nucellus, and this nucellus is the reserved food material storage, where they are having rich source of food materials. And inner to this nucellus is the embryo sac, where the embryo development occurs. And this entire structure is ovule, and this ovule, after the fertilization, it is forming into the seed. So this seed has various products. This seed has various components in them, like they have seed coat, which is formed due to this integuments, and they are having the endosperm, which is present within the embryo sac, and they are having the cotyledons. In case of dicot, it is two. In case of monocot, it is only one cotyledon, and they are also having the embryonal axis in the embryo formation. So this seed is the final product of the sexual reproduction. So seeds, they are the final products because the seeds, they are carrying the generation further. So once they are germinating, they will germinate and they are carrying the next generation forward. So that's why they are the final ultimate product of the sexual reproduction. So thereafter they germinate, they again go to the same cycle of germinate. They again go to gametogenesis, and then this gametogenesis again gametic fusion, and then fertilization, post fertilization. This cycle will repeat. So if you have to break the end of the sexual reproduction, that is only seed formation. The cotyledons of embryo. These cotyledons of embryo are, they are usually thick and they are swollen. You might have seen some of the newly growing plants, the young germinating plants. If you have seen them, they are some of the dicotyledons. What they have is they are developing the new leaves, and besides that, they are having the two fleshy leaves. And these two fleshy leaves that they are having is nothing but the cotyledons. And these cotyledons, they are thick and swollen, and they are carrying the lot of food materials for the developing or the uh, germinating seed. The integuments of the ovule. These are the integuments outer covering of the ovule. They are forming the seed coats after the fertilization. And the micropyle, this remains as such as an opening. And this opening is very, very crucial because it is allowing the oxygen and water while the germination is occurring. So for the germination to occur, they need some water. So water is the stimulation for the seed so that they can germinate. So that's why whenever we are sowing the seeds in the soil, we are putting the water. And this water is sensed by this micropyle opening. Through this, they are getting inside the embryo, and then therefore it is occurring the germination. So this micropylar opening is allowing the entry of oxygen. They are allowing the entry of oxygen and water, which is very crucial. That's why micropyle is playing very crucial important role. Like micropyle has allowed the pollen tube to enter to carry its male gametes into the embryo sac, and also now they are helping this oxygen and water to enter inside the seed so that there is a germination occurring. And these seeds, usually when they are young, when they are just formed, they are watery and they are having a lot of water content in them, and they are very fleshy. But as they develop, as they are maturing, as they mature, they lose this water content. As they lose the water content, they are desiccating and they are becoming harder. So as the seed matures, the water content decreases and the seed becomes very dry. And this dry seed is very useful because we are able to carry the seeds. We are able to eat the seeds only because they are able to mature like this and they are able to give us a lot of food. So the general metabolic activity of the embryo is slowing down when the seed is maturing. As the seed is maturing, the activity of the embryo means all the chemical activities, all the metabolic activities present inside, they are slowing down. So this is what we call it as dormancy. So embryo may enter into an inactive stage. That inactive stage is called as dormancy. So we have seen dormancy in case of animals. Even in case of seeds, this dormancy is seen where after maturation of seed, the embryo of the seed is becoming inactive. It remains in inactive period where it can be viable for various long time. Okay, that depends on the individual species. And there is one important terminology here. There is perisperm and pericarp. So they usually often asked in the exam, and we are often this we might get confused with these two terminology which are closely associated, and they are also resembling each other. So perisperm, some seeds they have remaining of this nucellus. The remnants of nucellus is nothing but the perisperm. The residual persistent nucellus is called perisperm. So example is in case of black pepper. In the black pepper, they have this nucellus present. We know that if you see here, nucellus is present around the embryo sac, and this embryo sac is developing into embryo. But this nucellus, when it is carrying this nucellus, the food storage materials as such, the all the food materials as such, even after the fertilization, such remnant nucellus or such persistent nucellus is called as perisperm. So in short, in order to define perisperm, it is a persistent nucellus after the fertilization. That is perisperm. So here you see in this picture, there is embryo and this embryo is surrounded by the endosperm. We know that embryo is present at the micropylar end and this is getting inside, this is surrounded by the endosperm and this perisperm. We know that perisperm is formed due to persistent nucellus. So nucellus is forming into the perisperm after the fertilization. Example for this perisperm is black pepper, and the next kind of word is pericarp. Pericarp. Carp is fruit. We know peri is outer, so outer layer of fruit. So example, if you see this picture clearly represents the 1, 2, 3, these are nothing but endosperm and they are having the embryo and then seed coat. Seed coat is formed due to integuments we have just seen, and these are all coming in the seed. But the fleshy layer, that outer fleshy layer which is present here is coming under fruit wall. So entire fleshy layer which is present around the seed, this fleshy layer of the fruit is called as pericarp. So pericarp is a fleshy layer around the seed. They are present in the fruits. So it is an outer layer of the fruit, that is fruit wall. Pericarp is a fruit wall. Perisperm is persistent nucellus. These two terms are very important to remember. So going further, the types of seeds. We have said that the seeds, they have some of them, they have endosperm. So some of the endosperm may be remaining even after maturation, even after development. Some of them, they might have utilized this endosperm. So based on this, there are two types of seeds, albuminous and non-albuminous. This non-albuminous also, they use the term exalbuminous, also they use this term. So albuminous seeds and non-albuminous seed. We are going to understand this terminology, albuminous and non-albuminous. Albuminous seeds, in some seeds, the endosperm is not completely utilized. The endosperm which is still remaining, such kind of seeds, they are called as albuminous seeds. So seed with the residual endosperm, seeds with the residual endosperm, they are called as albuminous seeds. And example for this is castor, sunflower, barley, and maize and wheat. So here there is a picture of the wheat. This is a picture of maize, corn, and the seed structure of the corn. We know that corn is a monocot since it is a grass. So it has this coleoptile, coleorhiza, and this coleorhiza is covering the radicle and the root cap. And on the other side, it is having the coleoptile, which is covering which is ensheathing the shoot apex, and the foliar structure. So this is a coleoptile, coleorhiza, and they are having only one cotyledon, that is scutellum. So that is scutellum, and this is entire embryo structure. And this, besides this embryo structure, they have endosperm. So from here, this is all covering the endosperm, and this endosperm remains even after the seed maturation. So even after the embryo development, after completing the embryo development, still there is some amount of endosperm present here. So this kind of endosperm presence even after the embryo development is called as albuminous seeds. So residual endosperm is called as the seed with the residual endosperm is called as albuminous seed. Example is maize, corn, or wheat, barley, or sunflower example. The next kind of seed is non-albuminous seeds. In the non-albuminous seeds, it is opposite to albuminous seeds, where here the endosperm is completely utilized or it is completely consumed. So the seeds without residual endosperm. So these are without endosperm, without residual endosperm. They are called as non-albuminous seeds. Example is pea, groundnut, and all the beans, the gram, these kind of structures. So where they don't have endosperm around them. So this endosperm is completely utilized during the embryo formation. That's why they don't have any more endosperm left over. So such seeds with no residual endosperm are called as non-albuminous seeds.